Good morning. Good morning, everyone. We are going to start the genetic basis summary. First of all, Dr. Ayuso apologizes for not being able to present today. My colleague, Dr. Fiona Blanco-Kelly, and I, Dr. Marta Corton, from the genetics department of the Foundation-IIS Díaz, are going to replacing her. In this session, we will review some basic aspects of the genetics of aniridia, and also we are summarizing the new perspectives that were discussed yesterday in the panel of genetics. As you know, aniridia is a rare disease with an estimated prevalence of 1 in 18,000 individuals, and it's a genetic disease. We have around 25,000 genes in our genome, but only one is causing aniridia. As you know, it's called PAX6. And all we have 2 copies or alleles in our genome, one in each chromosome 11 in a region called 11p13, and nearly all the patients, around more than 95% with aniridia, have a normal allele but also a mutated allele. PAX6 is a very important gene during ocular development for turning on other genetics during embryogenesis. And mutations in PAX6 cause and decrease production of the protein, and this is affecting the function of PAX6, and this is called haploinsufficiency. There are different defects in PAX leading to aniridia. Most of the patients carry punctual variants affecting the nucleotides, the letters of the PAX6 gene, but also we can detect, or we can find deletions in the chromosome 11 in around 20% of patients. As Dr. Sophie Vallée explaining us, she found in her cohort of French patients, and also we found in our cohort of Spanish patients. In deletions, the size and the involved genes influence the clinical presentation. If the deletions affect the PAX6 region, the patients usually manifest only manifest non-syndromic aniridia, isolated aniridia. But the deletions can also affect the next genes to PAX6. Sometimes some patients can have a deletion in a very close region affecting LP4. In this region, we can find the regulatory elements for the PAX6 expression. And in these patients, we have sometimes, or in several patients, a milder form of incomplete aniridia. But the deletions can be large. In this case, when PAX6 and the WGT1 gene are deleted, the patients can develop a more severe phenotype. This is called Ward syndrome. As you know, this is the acronym or tumor Wilms, aniridia, genitourinary anomalies, and a range of developmental delays. There is a full session about the Ward syndrome just in a few minutes. But also the patients can have extra large deletions that involves the BDNF gene. In this case, the patients can develop early onset obesity And this is a variety of the Warg syndrome called Wargrove syndrome. Most of the patients have punctual variants, and the clinical presentation depending on the type of mutation. The most frequent variant and the nonsense, that is, the change in the nucleotide cause and stop in the sequence. And then there is a truncation of the protein. Also, the patients can have very small insertion or deletions in the letters of the PAC6, and this causes a frame shift, and also a PAC6 truncating protein. In these cases, the patients usually develop a classical aniridia with high-risk and foveal hypoplasia. For the missense mutations, that is, the change of one letter for another cause a change in amino acid change in the protein, but the protein remains intact, is quite more complex. The missense mutations can lead for a highly variable presentation from classical aniridia, but also milder forms of iris or isolated iris or hypoplasia, no, high-risk hypoplasia, sorry, isolated foveal hypoplasia or optic disc hypoplasia, but also to more atypical forms of microphthalmia. And how we are doing the genetic testing? Usually there is a standard protocol, as I show you here, for aniridia, consisting on first the screening of the PAC6 nucleotides, the sequence of PAC6 by traditionally Sanger sequencing, but also the patient should be screened for deletions in the chromosome 11. There are several techniques. Before, or several years ago, the most standard was the chromosomal analysis using karyotyping or FIRS. Today, usually we analyze the DNA for the microdeletions by MLPA or microarrays. But we have to be aware that there are some limitations in genetic testing in aniridia. In a percent of patients, around 5 to 10%, the study is not informative. That is, no mutation in PAC6 is found, or we found a variant of unknown significance. There are some clinical and technical limitations. The phenotypic variability and the clinical overlaps can impair to identify a PAC6 mutation because other genes can be involved. And also, we need to use multiple approach to study the punctual mutations and deletions. Sometimes, not all the mutational spectrum of PAC6 can be done for the technical use. For example, is more difficult to analyze the non-coding or the regulatory regions. During the genetic panel, Professor Sophie Voller from the Hôpital Cochin in Paris talked about the new perspective in genetic testing. Some of the limitations I told you now can be overcome thanks to the use of the next generation sequencing that allowed in a single test for the screening of punctual variants in PAC6 and deletions. But also we can include in this panel for aniridia other genes involving in overlapping phenotypes. In general, we can use gene panels that increase the yield of the diagnostic in aniridia patients until 95%, more or less. And we can also find other genes or mutations in other genes, such as FOXC1, PIC2, and as the Professor Valle reported, C1b1. Genome sequencing is a new promising technology that can help to detect structural variants in PAX6 negative cases. She has more resolution to detect, for example, inversion. We discussed several cases in this panel. Then, Dr. Alexander Dransoni from University Hospital of Udine discussed in her talk another interest aspect of how functional in vitro analysis can help to understand if a variant in PAX6 can be or not pathogenic, and then it could be or not the causal genetic in the patient. There are a few variants of unknown significance in PAX6. It can be mainly missense or splicing variant, and also in the non-coding regions. And in vitro analysis can help to model if they can affect the RNA process or the function on the PAX6 protein. And other interesting talk in this panel, Dr. Elizabeth Simpson from British Columbia University in Canada introduced a very promising strategy for genetic correction of PAX6 mutations based on CRISPR-Cas9 and using a new delivery system into corneal cells using nanoparticles. With this novel approach, she and her group were able to correct and PAX6 a nonsense mutation in the corneals of the mouse model small I after an intrastromal injection. And now my colleague Fiona Blanco is going to resume other aspects in the aniridia genetics. Yes.
Hello. Hello, my name is Dr. Fiona Blanco Kelly, and what I'm going to do is a brief summary of genetic and reproductive counseling. So a little bit, all that Dr. Corton told you, how we manage it at a clinical setting. So I'm not going to go through genetic aspects because my colleague already did it, but what is all of this for? Why do we do genetic tests? Why we improve, or we get to try to diagnose every single patient with their genetic specific alteration? Well, this is to confirm the diagnosis. It's basic to know whether there's or not risk of Wills tumor, also for prognosis and management. That's very important for patients. And for reproductive risk, to know whether I have a risk of having an affected child again or not, to avoid transmission, if I want to do that, and of course for future perspectives and treatments. So in a clinical setting, it's very important to take into account when assessing and testing the patients, their age, the age of the parents also, whether the mutation has been identified or not, whether the technical feasibility of detecting it regarding the technique used, and the type of mutation or pathogenic variant that we have detected. Also, it's very important whether it's de novo or inherited, and whether it leads to an isolated phenotype or a syndromic phenotype. We all know that familiar aniridia is an autosomal dominant condition. So that means that the affected person has a 50% risk of passing the genetic alteration and therefore the clinical, the aniridia. And when we have already detected the mutation in the family, we can open many doors for reproductive assessment, where we have pre-implantational genetic diagnosis, prenatal invasive diagnosis, and non-invasive diagnosis, that we will go through it further on in the presentation. And don't worry, when we don't have the mutation, we can still do things, we can still help, and we can still research and try to find the cause in those families. The other main cases in aniridia are the sporadic cases or de novo cases. Here we know that from the patient on it will be like a familiar case, but we also have the parents that are always a bit worried about the risk of this happening again. Of course, the rest of the family has the population risk for aniridia. In sporadic cases, when we don't have a family picture that we already know whether there's likely to be a Wills tumor or not, the first thing that we have to do is to rule out WT1 deletion. And in this sense, what we are ruling out or trying to find is whether the patient has a syndromic form of aniridia or not. So whether they need further follow-up apart from the eye or just the eye. So as it happens with the familiar cases, when we know the mutation, we can provide the patient with reproductive assessment, and we can also provide assessment to the parents, because we know that the risk of recurrence is very low, but it's not zero, and we always doubt about mosaicism, which means that maybe some of the cells in one parent do present the genetic anomaly, but as it's not in all of their cells, and maybe it's not in the eye cells, they don't manifest the disease. So for the patients of a de novo sporadic case, we can do prenatal invasive diagnosis, also prenatal non-invasive, and it's very important in these patients, although this is not currently, we are not so aware of this many times, is that when we find microdeletions, it's advisable to perform a very classic technique, which is a karyotype, to determine whether there's a rearrangement on chromosome 11 in one of the parents, which would change the recurrence risk. And this is not to do with mosaicism, but with chromosomal alteration. And of course, we always also have research for these patients. As new techniques are being developed, now we are able, in some cases, to detect the presence of mosaic parents in some aniridia cases. Well, as per reproductive assessment, most of us know about a prenatal invasive diagnosis, which is amniocentesis of a chorionic biopsy. It's while I'm pregnant, we get cells from the gestation tissue, and we can study the karyotype and also the DNA, which means we can study PAC6. Then we have non-invasive prenatal diagnosis. This is we don't need to perform an invasive punction on the pregnancy tissues. We just get blood from the mom, and we try to detect the mutation in her blood, whether it's present in the fetus or not. This is an ad hoc kind of study. It has to be designed specifically for each patient and each family in order to be sure that we don't detect what we don't have to detect, and we detect what we have to detect. In our experience at the Fundación Jiménez Díaz, it's been already around 70 cases, including aniridia patients, and we've been doing this technique since 2005. Then, the limitation of this technique currently, it's because that we cannot do it for all of the mutations, and as I said, it has to be designed by patient, family, and mutation, and it's tricky yet for maternal mutations, but this is, we are getting there, and said by Dr. Corton, new technologies just not allowed for detecting unsolved cases, but also it allows to detect or to rule out maternal mutations in pregnancies of aniridia patients. And then we have the pre-implantational genetic diagnosis, which is a process made for embryo selection. So this is a quite a complex process in which we have to go through many different parts or items that have to be fulfilled in order to be able to perform this kind of study. The inclusion or exclusion criteria include genetic, legal, and gynecological. Genetic and gynecological, andrological are the same across countries, but legal may change. Depending on the country you are, the access to this technique may vary. So PGD, basically it's we create the embryos in a lab, we biopsy them in day 3 or 5, depends on the department, and we study them. And as you see in the picture, we don't just study them for the mutation, but also we try to mark the chromosome that carries the mutation, so we are able to clearly distinguish between an affected embryo and a not affected embryo. This is very important. It's called informativity studies, and generally requires to test not only the patient and the partner, but also the parents of both of them, in order to be sure that we don't make a mistake when we are selecting an embryo. So when an embryo is fine, it's free of the disease or the genetic change, we can transfer it so the woman gets pregnant, or if we don't want to transfer it, then we can just vitrify it and save it for later on. This is how we do things at our hospital. As you see, there are quite a number of steps that we have to go through and fulfill in order to get to the next step. That's why, although many people begin this process, many they cannot fulfill and get it done. We have been doing PGD since 2008, and we have performed already nearly 1,000 PGDs at our hospital. So thank you very much to all of the patients, the scientists, the organizers, and to my colleagues at home that do all this work with me every single day. Thank you.